Okay, after we've learned how ERPs are calculated, I would like to show you a few examples of how ERPs were used in actual psychological experiments. For example, in this study by Roncion and Jacques, there were faces and cars presented to participants, and they had to indicate whether the object was facing to the right or to the left. And what the researchers observed was a distinct ERP pattern at around 170 milliseconds here, and that's why they named it N170. So there's different ways of naming ERP components. The P1 is the first positive deflection, N1 is the first negative deflection, but sometimes they're also named after the latency. So the N170 is called that way because it is maximum at around 170 milliseconds. So of course it's great to have a marker for faces, because sometimes you might be interested in isolating the processing of a face and how face processing varies between participants, between maybe emotional states, etc. So you need to make sure this component is really related to face processing rather than other things. So could it be that there's a confounding variable here? For example, it might be that the faces are just more familiar stimuli compared to cars, and this might actually affect the lateralized the deflection in the ERP. To test this, Bantin et Duel did this kind of experiment where they compared the N170 between familiar and unfamiliar faces, but it was actually the same. Only later there was some difference, but the N170 was not affected by the familiarity of faces. So that's one confounding variable we can exclude. And of course there were similar experiments done like that. Once you discover a new ERP component, there's of course a lot of research trying to pinpoint at the actual functional significance of that component to exclude any potential confounding variable and enabling researchers to use it as a reliable tool to look at specific cognitive processes. Here's another study that used the N170 by Tanaka and colleagues. They compared participants' own face, a familiar face, and other faces, so non-familiar faces, and they found again the N170 was not affected by own face or familiar face, whereas the N250, a bit later, is actually reflecting own face, familiar face, and other face. So this is often what you find in ERP experiments. You look at where do we find the difference and what does that tell us about the cognitive processes. Another example of where ERP components are particularly useful in helping understand psychological processes is the P300 or P3. Again, P300 refers to the latency, P3 refers to the order, so the third positive deflection. And there's different schools. Some people prefer this nomenclature, some people prefer the order nomenclature with P3. Either way, we're talking about this component now. You will often hear P300 because that's how it was called initially. And just to explain you how useful the P300 was for this debate, you don't really need to understand the implications of this model right now. All you need to know is there were competing theories on how and when the brain selects which information from the vast input of information is processed. The early selection models claim we have a sensory store where then based upon the sensory store there is a selective filter letting only one thing through, and this then reaches high-level processing. The late selection models had a different view. They basically said that there's some higher level processing first, so all the information goes all the way through to higher level processing, and only then information is selected. So that of course means that here the selection is based on sensory sensory features only, whereas here the selection can also base on some higher level processing. Now the problem was that both models had some good evidence, and there was like a long-standing debate that was, however, pretty inconclusive. That's always a little frustrating for experimenters. Of course, they want to know the truth. They want to find out, is selection early or late? And with purely behavioral measures, that was just very difficult to assess. But then came along Stephen Hilliard, one of the pioneers in ERP research. He used the P300 component to solve this debate. So in this task, participants wore earphones, and they heard tones on both the left and right ear. So in this condition, condition A, participants had to only respond to high tones on the left ear. They could disregard all tones in the right ear altogether, and they could also disregard low tones on the left ear. So this would be the only target stimulus in this example. And then they had to count how many of these tones were presented. In condition B, it was just the opposite. Now they could ignore the left ear and only had to count targets on the right ear. Again, high tones. So how does this experiment help us to resolve the question whether processing of the whether the filtering happens early or late? Well, one thing you need to understand here is that in order to understand whether it is a high or low tone, you need to decide. So you need to decide whether it is a high or low tone. That needs further processing, higher cognitive processing. That should be reflected in the P300. Whereas early filtering would indicate that you can actually tune out, for example, the right ear altogether if you're only trying to listen to the left ear. So filtering is happening on the level where you can just focus on one ear and then disregard the other ear. Whereas target identification is what's happening in each ear. So you need to tell apart the target tones from the distracted tones. So how does the ERP respond to that? Well, the N1 component that is related to sensory gain is reduced from around 80 milliseconds on. So if they attend an ear and there is a stimulus, then you can see there's a higher amplitude compared to here. This is not just targets, this is just all stimuli altogether. And here you can see the opposite. If you attend the, this is left ear stimuli, so you should attend, when you should attend left, then you can see the N1 is more pronounced. If you're supposed to attend right, then it's less pronounced. So in sum, whenever there's a stimulus on the ear that you're attending, there is a larger component. So this suggests that there is already an early filtering going on at around 80 milliseconds. Now the question is this before or after participants have higher cognitive processing, so before or after they can tell apart targets from non-targets? And it is actually, the filtering is happening actually before, because here the P3 or P300 you can see is only appearing for task relevant tones, for targets. This is the target, this is the target, and the dark line here, that's the what they call standard or non-target or distractor. That's the tone you can ignore. So there's differences between target and non-target tones from 250 milliseconds on, clearly after the filtering. And this requires higher processing. So the filtering is happening before higher level processing is necessary. And this is of course evidence for the early selection models. Another component that is interesting for psychologists is the N400. This is particularly interesting if you're doing linguistic research. So the N400 is related to deviations in the semantics. To give you an example that makes it a bit clearer, I think, just read these sentences by yourself. Okay, first sentence, second sentence. Okay, you can see, compared to the first sentence, there was a semantic incongruity. You wouldn't expect someone to spread the socks on bread. Last example. So here you can see a physical deviation. The last word was just larger than the previous words. So how does this affect the EEG signal, and what does it tell us? So here you can see one trial with the onset of individual words. Of course, this is a little bit all over the place because this is just random processing of different words. But the critical part is really the last word, because here we have a semantic incongruity or physical deviation compared to the standard, which is this black line here. So if there's a physical deviation, you can see this positivity. If you see a semantic incongruity, so spreading the warm bread with socks, a word that you wouldn't expect, then you see this N400 here, the negativity 400 milliseconds after the onset of this word. And the interpretation of these results was that there's a violation of the semantic expectancy. So basically the idea is that you build up some expectation of which words would likely come, and then if another word comes, this needs further processing to implement that into the semantic processing, and this effort is reflected in a deviation in the ERP component. The N400 is also affected by the semantic relationship. So, for example, if there is, so this is a typical member of a. So imagine you have a word that is, imagine you have a sentence, and the last word of that sentence is, given the context, likely going to be a bird. Then, if a typical member of the bird, in this case it's a sparrow, comes, then you can take this as some kind of standard condition. And if you compare that to an untypical member, for example, you expect a bird and then a turkey comes, then you already see some deviation towards the negative. So this is some deviation of the expectancy, but not as much as a completely unrelated word. So this is the classical N400. And then if you have an antonym, that's even more positive than the standard. So what this means is that the N400 seems to track the effort to retrieve the stored conceptual knowledge. So basically, when you're processing this, you're expecting a certain word to come, and if it comes, then you have a little effort processing that word because it's already pre-activated. If it is an untypical word and you need to retrieve it, there's some effort, for example, the turkey thing here. And if it's a completely unrelated word, it's even more effortful. So the idea is really that the preceding words can serve as a cue and make retrieval easy, and that's really how language processing is working according to various theories. The N400 is also independent of sensory modality, so it doesn't matter if you read it on the screen like we did in our example here, if it's spoken to you, somebody reads it out, or if it's a line drawing, like a picture. This is also illustrated by some work that Melissa Vaux was doing. She calls this scene grammar. So when you see this scene where a mouse is at the right place here for right-handers, I guess, on the right side of the laptop. And then in another scene, you have some kind of soap dish next to the laptop, which is not where we would expect it. And then you can also see this in the around N400 range, that this is more negative than this. Again, hinting towards the notion that you need more effort to implement that into your scene, rather than here where it is congruent or consistent. So much about the N400. So far we've talked about unlateralized potentials. So that means you look at a single electrode or a pool of electrodes, like for example these 3 electrodes, there would be a pool, so you can just average across these electrodes. And this is irrespective of the laterality on the screen. So this is unrelated to where on the screen something is happening. Now, a lot of experimental questions we have in psychology are related to the lateralized processing. So, for example, when you see a target on the right hand side of the screen, then there is actually a lateralized processing of that stimulus. So we need to also look at lateralized potentials in the EEG signal if we want to fully exploit the EEG data and gain as much knowledge as possible. So how do we do this? So in this example, if the target is on the right hand side, that means that the contralateral electrode, for example, would be the pt.2, and the ipsilateral electrode, same side of the target, would be the pt.8. And then of course, if you switch the sides, and it's the opposite, so you can do this for electrode pairs, and then you end up with something like this table. Just shows you what contra and ipsilateral means, and you could calculate this for pt.7, pt.8, and then maybe end up with something like this. So here you can see that there's not really any difference. There's no lateralized processing up to this point at around 200 milliseconds, and then contra and ipsi deviate, and then it goes back to being aligned again. You can also show the same data as the difference wave. So this is the same, really just showing contra minus ipsi. And then this little bump here, that's the N2PC. It's in the N2 range, and it is a posterior side. So for example, pt.7, pt.8, and it's contralateral, hence N2PC. We'll talk more about this component in the attention session. For now, you just need to understand that it is useful to use lateralized potentials here because they tell us something about how the target is processed. And another advantage is also that, remember, there's always some noise, and if we calculate the difference between contra and ipsi, we're subtracting any kind of noise that would be affecting pt.7 and pt.8. So imagine, for example, there's some kind of, say, movement of the head that affects pt.7 and pt.8 the same way. There would be noise on each of these, on the red and also on the green wave, but by subtracting ipsi from contra, that would also cancel each other out to be 0, and then we end up with a smoother signal in the difference wave. Another lateralized potential is the LRP, the lateralized readiness potential. So in the previous ERPs we've talked about were always related to the stimulus onset. So event-related potential always means it is related to an event, but the event doesn't necessarily have to be a stimulus on the screen or on your ears. It can also be your response, for example. It just has to be a well-defined event that ideally happens in every trial. So if you have a response by the participant, you can also average across trials and take this as the point 0 in each trial. And then what you see in the lateralized readiness potential is just before participants respond, they have this positive going deflection in the ERP. And this is related to the preparation of motor activity. So this component is also strongest near the motor cortex. It is easy to isolate from other ERP components because of the location and also because it is related to the response but unrelated to the stimulus presentation. Here you can see an example where the LRP was used in Kato and colleagues 2009. They had a go, no-go task. So a go, no-go task is a task where you only respond to one stimulus and you just omit the response for another stimulus. They made this task particularly boring and tiring and also long, 1,440 trials. And what you can see here is that the component LRP is basically a function of mental fatigue. That means if you compare the first interval, the first third of the experiment, you can see that the LRP is most pronounced, and then in the second interval it's reduced, and in the third interval it's even more reduced. And what does it mean if the LRP decreases? Well, it means that the response execution is less efficient. So it seems this experiment suggested that the accuracy that decreased throughout the experiment, that was behaviorally tested, is due to mental fatigue because participants are less efficient at preparing their response. So this is a way to look at motor processes and response preparation, for example. Okay, so you've seen some examples of how ERPs are used in psychological experiments. There will be more examples in the individual sessions where you can see how they're, for example, used in working memory and attention. Next and last part of today's session will be about advanced EEG methods and MEG.